talk that will be presenting the Omics Logic platform. We will have two persons that will be speaking today to us. Allow me to introduce them. I am happy to introduce the CEO of Pine Biotech, Mr. Elijah Brodsky. He is one of the co-founders of the Pine Biotech company. Elijah's background is in applied mathematics and data science. He is currently completing his degree in business and management from Tulane University. He has facilitated and managed collaborations with researchers at Stanford, Louisiana State University, Tulane, and other academic and pharma R&D teams. Mr. Brodsky oversees the strategic development of research projects and manages the team responsible for bioinformatics analysis, training, and collaborations for Pine Biotech. Dr. Mohit Mazumdar has over 12 years of experience in research and industry. He completed a master's in bioinformatics from Jamia Millia Islamia and a PhD in computational biology and chemistry from Jawaharlal Nehru University. He is an expert in machine learning in application to biomedical and chemical data. He is currently heading the global business development for Pine Biotech USA and contributing to bioinformatics research and education by collaborating with universities in India and Africa and mentoring participants from various programs by providing bioinformatics research, supporting applying statistical and algorithmic approaches. We thank the sponsors for this activity, Puerto Rico INBRE, University of Puerto Rico Medical Sciences Campus, and NAIPI, Omics Logic, PR INBRE, and the Talbot Bioinformatics Research Center. Please, Elijah, proceed.
Okay, thank you very much for the introduction, and thank you everyone for having us today. I want to start by briefly sharing some background information about NAIPI. For those of you that are not familiar, NAIPI is the National Association of Idea Principal Investigators. And as a part of this organization, it connects the idea community. So the idea community are the states, 23 states, including Puerto Rico, that are funded through the NIH to elevate the level of biomedical research. And you can find some more information about this organization on the website, naipi.org. And thank you. And I'm here presenting today's session on behalf of NAIPI, essentially because some of the materials and resources that we will cover today are being offered through NAIPI. And so to briefly explain what NAIPI's objective is in this program, I will also share maybe a link, or Jesse, if you could share the link to the NAIPI website that explains essentially what our program is designed to achieve, which is provide training and link resources across this network to offer elevated access to biomedical research using computational tools and bioinformatics. Okay, let me start my presentation. Okay, so what I want to share with you today is the project-based training in bioinformatics and data science that we have designed to address a gap in skills and resources that we think exists across these 23 states and Puerto Rico, primarily starting from the undergraduate level and offered as an integrated path from learning to research. And so before we begin talking about what exactly this program includes, I want to introduce you to a portal that we designed to make sure that these resources are widely accessible and effective using our asynchronous portal called Omics Logic. So if you please go to learn.omicslogic.com, And I believe that the link to the portal will also be available in the chat. You will find here that, we explain kind of what this portal is all about, biology as a data science, on the front page. And I will walk you today through some of the resources that this portal includes and how this activity can be used for training and preparation of students for research activity using bioinformatics and computational biology. And then later on, I will pass it on to Dr. Mohit Mazumdar, who will speak about some of the ongoing programs that we currently have offered through the Louisiana Biomedical Research Network and now being available to the NAIPI community, as well as some of the upcoming events and resources that are scheduled for the spring and the summer of 2022. So if anyone wants to understand how this portal works, it's always useful to start with creating a profile. So you will see that there's a login button, and it's very easy to create an account. And so I will encourage you to just try it out yourself right now. If you can go and create an account right here, as you click on sign in, it's a free sign in. You then create an account by clicking this button. And the first thing that you will see is that everything that we will be discussing today is focused on that user profile. So you'll be able to see how this portal works, and I will introduce some of the elements that we use in training for different types of omics data analysis and data science. So once you log in, you will notice that there is going to be a user profile. So right here on the top, you will find your email address. And in that email address, you will find all of this activity related to your user profile. And we use this information in part to validate that the user participants are a part of the NAIPI network, because for them, all of this training is available for free throughout 2022. So I'll give you just a moment here to sign in, and I will switch over to my own account just to illustrate how this works. Okay, so learn OmicsLogic.com. If I log out, you will see right here on the top of the screen, you will see a login button. And here you can create an account. And you can either use your email and create a password, or you can sign in with one of your social media accounts. So I already have an account. I'm going to just sign in. And after you're signed in, you will be redirected to either different levels of access, so you can skip that, or to this part of the portal that lists out the different coursework that we have. And so today I'll briefly introduce some of these resources, but I wanted to start from this overview of your account. So on this account, you will find your personal activity and certification as you complete some of the coursework. So what we've realized working with the Louisiana Biomedical Research Network, as well as with other states, is that expertise in bioinformatics is really not distributed equitably or equally throughout the different regions where we interact with students. In fact, what we've noticed is that there's a big gap in even understanding of the role of bioinformatics and data science in many aspects of biomedical research. And the challenge, I think, is that we don't introduce these concepts early enough in the kind of horizons of the students that are joining research projects. And so as a result, there's a gap in skills. When those students want to participate in meaningful research, you want to involve them in a research study, in a grant proposal, or collaboration, they are not equipped to deal with data. And so to introduce different aspects of skills, including technical skills as well as conceptual skills, or what we call the logic behind omics data, we've designed this portal that seeks to address the gap in skills and the understanding of how biomedical data could be analyzed to produce meaningful insights. And one of the things that we are working on is a certification provided through regional as well as local institutions, For example, LBRN, the Louisiana Biomedical Research Network, would be a regional institution, and LSU, Louisiana State University, would be a local entity. They can provide training certificates that validate that the student is ready, or a researcher essentially is ready to participate in all aspects of research that include bioinformatics. One of the ways that we do this is through our tracking system that looks at different aspects of participation, activity, and skill sets that are covered in the different topics that are introduced throughout the portal. An important component of that is a project. So a lot of times when we try to understand if a student is ready to participate in a project, it's not enough to just have basic skills like coding in R or statistical analysis. What we really want to understand is whether a student is ready to understand the question, the research question, and the approach that could be taken using data to address that question. And so a big part of what we are trying to do is design a system that leads from basic training, which is skill-based training, to research-based training, which is project-oriented. And so this project process that we've designed here starts from completing your profile and presenting yourself to the world, that goes to earning a certain number of points, which validates that you have the technical skills. And then eventually it gives you access to a specific course that addresses common challenges that people face in designing and developing an independent project using bioinformatics and computational biology tools. So as a result, what we see is that those students that are able to complete a project, and here we have, sorry, I'll go back to here. Here we have an example of what this project structure looks like. You can see that it actually prepares a student to think about their research presentation for a poster session or even a publication. And I think that's really a critical need that we all face in giving access to research projects and involving students, especially undergraduate students, in biomedical research that is ongoing, right? We want them to understand the format. We want them to understand reproducibility of research, and we want them to be able to describe the methodology and the approaches that they take in technical language that is brief and something that we can all review. So that is an essential part of the training that we provide. And to be able to get to that level, to be able to address kind of the place where students commonly are when they begin this training and where they should be when we anticipate them to be ready to participate in meaningful research, we provide a curriculum that is organized by several different subjects. So first of all, we have different topics that we cover, including methods of analysis, for example, statistical analysis on machine learning, types of data, genomics, transcriptomics, epigenomics, and metagenomics, as well as different levels, for example, getting started or trying to address a skill in R coding or Python coding, and eventually getting into a project. And projects here refer to publications that are digested, curated data sets from that publication are extracted, and reproducible methods of analysis that follow the paper method section are given as a tutorial. So to get started with these, we organize a getting started or introduction to bioinformatics curriculum that anyone can try, use for themselves, or utilize for their training and introduction to students that don't have even the basics of bioinformatics as of now. So the introduction to bioinformatics consists of several different modules. They're all organized under this Getting Started section, and they include Introduction to Bioinformatics, which is a course that speaks about the applications of bioinformatics in different domains like healthcare, pharmaceutical industry, basic and translational research, environmental research, and others. It also covers the basic terminology that connects biological concepts and data science concepts or information technology concepts in a course that we call Bytes and Molecules. So the course is designed to introduce terminology and the basic concepts that one has to know to be able to continue and pursue bioinformatics or computational biology as a practical skill. And then we offered 2 simple courses in bioinformatics in R and bioinformatics in Python that are designed to show how easy it is to start with coding for someone that has minimal or no background at all in coding, and has some immediate applications to the types of challenges that one might face dealing with biological data. Now, apart from that, we will talk today about several other resources that we utilize inside this training that help build the logic of analysis focused on big data. So we are following this specific 2-directional approach. One direction is practical skills, and those include concepts, terminology, understanding, critical thinking. And then we also introduce this other dimension, which is the logic of analysis and how to apply some of these concepts to large data sets that typically you would not be able to handle on your own system. And so those technical limitations are eliminated on our Bioinfo platform that I'll discuss in a moment. Importantly, like I mentioned, all of these topics are introduced in the context of projects, right? Because one of the things that we realized is that it's very difficult for somebody who's interested, let's say, in infectious and communicable diseases to understand the specific applications of bioinformatics in a context that is completely different, let's say in environmental data or maybe cancer data, right? So we want to make those applications as closely related to the type of interest that the student might already have. And so we organize our different tutorials under projects, and those projects could be divided into different sections. So what we started with this year is offering 6 basic training modules. And these 6 basic training modules are Introduction to Bioinformatics, Genomics, Transcriptomics, Metagenomics, Epigenomics, and Data Science. So these are offered as year-long curricula that you can organize into an asynchronous or a live type course. Our team supports it and actually runs a number of these programs throughout the year. And Dr. Mazumdar will be speaking about some of these programs when I'm done with my part of the presentation. In addition to that, we also provide specialization tracks, and these include oncology, infectious diseases, neuroscience, and agriculture. And agriculture includes also some elements of biotechnology. So what we are trying to do is develop exemplar projects with curated data sets and an explanation of methods that are sourced from impactful publications in that domain to illustrate how some of those basic data types as well as methods could be applied to solve real research challenges. Now, let me briefly explain what is the difference between what I am describing as a curriculum or as a content database and some of the practical resources that are included in this program and in this training. So the OmicsLogic program resources are really designed around training materials, computational tools that are easy to use that biologists can use, interactive coding exercises that could be, first of all, checked for syntax, and second of all, checked for the correct results. And then finally, an introduction to a coding environment where you can take some of the scripts that you've learned how to write and then execute them on a big data infrastructure. So let me briefly introduce some of these resources in greater detail. One resource that is very critical for a biologist to get started is no-code bioinformatics. And that means that you can use bioinformatics without writing a single line of code. And for that purpose, we have a big data analysis platform that is not unlike Galaxy and some of the other tools that exist out there, but it is tightly integrated with the curriculum that we have designed for different types of data. And I'll briefly show this platform today so that you can all get a sense of what it really looks like and how it works. The second part of this tutorial approach is incorporating data science, and that means coding exercises that check your ability to write correct syntax without errors and provide you with specific results to the coding challenges so that you can understand how to get the desired outcome in your code following a tutorial. And then finally, we also are building a network of mentors that can help you take those skills further. And so essentially by working on exemplar projects and trying to identify research questions that could be answered with data analysis skills, we can then eventually match you to a research experience in which you can try to understand a topic with the help of a mentor that will eliminate some of the dead ends and guide you to identify a low-hanging fruit in terms of being able to answer a research question as an experience for you to gain computational biology research skills. Let me briefly show you the resource that I mentioned before. So right here, if you go under courses on the portal, and by now I hope that most of you have already created an account. I saw that some people joined maybe a little bit late, so I'll just remind everyone. This is a portal that we are speaking about called Omics Logic. So you can log in by following the link learn.omicslogic.com. Login is free. You can gain access to most of the resources for free. And if you are a part of one of our programs through the NAIPI, the National Association of Idea Principal Investigators, you can gain access to all of the resources that I'm mentioning today for free using the NAIPI contract that our company has. So here you will find different types of courses, materials, and projects. Right here, you will find different tags that you can try. And below are collections that you can find focused on different types of data, different types of skills. And one of those skills is the Bioinfo platform that really establishes the logic of analysis applicable to big data. So what is this platform? This platform, and by the way, anyone that wants a demo account on this platform after you have registered on the Learn portal and you have received your password in your email, after 24 hours, you will gain automatic access to this platform just to explore and see what it can do. So this platform is organized according to several different sections. You can see here transcriptomics, genomics, epigenomics, and different methods of analysis listed on this side. There's also sections related to mass spectroscopy and structural biology. There are specific sections and application to cancer biology, single cell analysis, virology, as well as data integration and modeling. So as you look through these different sections, you'll find that some of them have a drop-down button. And this drop-down button gives you access to a demo pipeline builder that you can try without having to upload any data and without having to perform the actual analysis. So here, for example, you will find that informative pop-ups that are associated with each step of the analysis will guide you to build meaningful combinations of scripts and algorithms that will produce results that you can use. So for example, in this transcriptomics section, you can see that I can perform mapping using bow tie and then visualize the SAM file of the mapping that I performed. And in addition to this visualization, I can also find resources on the Learn portal that will allow me to understand what exactly I am trying to do. So this is just an example. Again, you can see that in many sections, there's a drop-down that you can find different types of methods of analysis and different types of pipelines that you can perform. So you can try and use that demo account once you have created a login. So again, that could be found using coursework on Bioinfo, and you can see here a collection of courses that you can explore and really learn how to perform analysis of metagenomic, genomic, transcriptomic, and other types of data. Now, to illustrate how these kinds of methods are used in a training experience, I want to briefly talk about several different types of programs that we run throughout the year. And then I will pass it on to Dr. Mazumdar to speak about specific programs that we have running right now, starting in the end of January. So a good example of a program that we regularly run is a program on transcriptomic data analysis. And so we call this bulk and single cell transcriptomics. The program is designed to introduce the omics technologies, including how the library preparation happens, how the actual sequencing happens, how does the understanding of the methods of data preparation impact our analysis approaches to that data, and ultimately how to then use some of this methodology to actually be able to perform large-scale analysis using these kinds of methods. So again, starting from the Bioinfo platform that you just saw, we will go through some example pipelines that students build. They build those pipelines to understand the inputs and the outputs behind each step, as well as the logic of analysis. How can they apply the right combination of scripts and methods to achieve the desired results? And all of it is very intuitive, right? As you can see, there's no coding involved. In addition to that, the platform is color coded, and this color coding follows a logic that we explain in detail throughout the training program itself. To apply those methods, we then introduce several different projects. For example, cancer macrophages, modeling cancer precision medicine, PDX tumor in the microenvironment, but also elements of infectious diseases like malaria infection, or even applications in biotechnology and agriculture like potato drought resistance for modified crops. So that gives you an understanding of the specific skills or specific methods that need to be used, and the specific applications where you can actually try and run that kind of analysis and see what are the results, what type of insights you can generate analyzing such data. In addition to that, we then go on to one of the maybe most exciting areas of transcriptomic data analysis, which is single cell RNA-seq analysis. And we also have a very basic processing pipeline, as well as a downstream analysis pipeline for Seurat and Monocle, that we go from the logic and then apply all of the logic in terms of code that could be run on Google Cloud Platform using Google Colab. So what happens is that you understand the logic, the need, and the reason for specific methods, and then you can apply the specific portions of that code to try and modify and get the types of figures that you might need for a publication. Moreover, you will then see how the exact same approaches are used in high-impact publications that are really transforming our understanding of different domains, from cancer to neuroscience, using such techniques. So again, the objective in such a program, in such a basic program, is to introduce the specific type of data, the methods that are typically used to analyze such data, and then showcase some of the applications of that data analysis, including multi-omics integration. Another type of a program that I want to briefly speak about is bioinformatics for infectious diseases. So one of the things that really happened over the past couple of years is that even people who had no understanding of mutations, variants, evolution, and things like that really now know this terminology and are even interested to try and ask questions about the huge amounts of publicly available data related to the pandemic, but also as an extension of that related to many other types of epidemics or outbreaks that could be occurring. And so what we did is, a couple of years ago, we worked with the virologists here at the Louisiana State University, understood how their expertise in virology and bioinformatics could be used for creating these training modules, and then continued our collaborations with researchers at UCSF, Stanford, Louisiana State University, Georgetown Medical Center, as well as other regional centers, for example, a large research consortium on malaria, to create project examples that illustrate how similar types of analytical methods could be applied to analyze data and understand these outbreaks of infectious diseases. So in this program, we use project examples related to the enterovirus EVD68, which was an outbreak that affects primarily younger children and happened recently in Europe as well as in the US. Malaria infection, again, endemic in Africa, but also in many regions of South America, I believe, and in Asia. SARS-CoV-2, which we all are continuing to observe, as well as the Ebola virus epidemic that not only started the first public and connected collection of next generation sequencing data that is publicly available, but also produced solid results, including vaccines and antiviral drugs that treated a disease that was 90% deadly before that. So as an extension of that, we also offer a focused analysis of SARS-CoV-2 as a training module that many of the research institutions that we work with can use immediately, because a big part of the sequencing and a big part of the research questions around SARS-CoV-2 are regional. And so by working with institutions like Louisiana State University, where we did many different projects on SARS-CoV-2, including wastewater sequencing, we can train students and researchers to be able to perform the same and ask questions of the publicly available data. And so on the Learn portal that you have just seen, you will see this kind of an overview that gives the basics of multiple sequence alignment, as well as the ability to ask questions about origins of SARS-CoV-2 or how the new variants are emerging and how they might affect the future of this pandemic. So at this point, I briefly want to introduce another element that I think is critical for those students that are serious about their interest in bioinformatics. And what we see time and time again is that students going through short-term training are unable to capture a lot of the aspects of bioinformatics, the application of bioinformatics, but also the ability to independently learn and continue to apply bioinformatics once those resources are unavailable. And so for that reason, we wanted to make sure that many of the same methods that are available through such training could be taken away with the researcher or the student after the program completion, and they're able to replicate and even use those in their reporting or ability to analyze data. And so I briefly want to show you a course that is designed to kind of give you a broad understanding of how this portal works. And that will be Introduction to Bioinformatics. So if you go to Introduction to Bioinformatics, bytes and molecules is what I wanted to say. Okay, so bytes and molecules. The Bytes and Molecules course really covers many of the aspects of what we just discussed for someone who is a beginner and has minimum or no background in bioinformatics, wants to understand the terminology that is used in such training, and wants to be prepared with some of the technical skills that are going to be needed. And one of the courses that will be available in this section is an introduction to coding. So coding is an important part of a bioinformatician's day-to-day life and activity, but it is often seen as an exclusive skill set that only bioinformaticians and engineers really need to have. But as we all know, in many aspects of research, just to produce the picture that you need, the figure, maybe some simple data analysis with some statistical properties, it is always useful to be able to know that piece of code that if you know how to run it, you know how to give it the right input, you can make it produce the right output that you want. And that is exactly the objective of some of this very basic introductory training that we offer in this section called Introduction to Bioinformatics Languages. And this one is an example using R. So here you will find some very basic things that you need to understand about data, how it is stored and how it is used in the coding environment. But we don't have to set up a whole environment on your own system to be able to run and produce even the basic plots that you might want to use in your research publications or exploratory analysis of a data set that could be very informative. So we offer here inside the actual lesson a console that you can run and get result right here. And as you can see, immediately, if I just run this piece of code, I am getting an error. And the reason is because obviously this path to the file does not work, right? And so one of the basic challenges that all of us probably faced when we started learning how to code, I can learn how to write hello world, but how do I actually make this code work for my data, right? And that's exactly where we start. So just by copying this link and replacing it right here in the console, I can run the code and I can get some results, right? And so right here, you can see that I can check the result. And now I have a successful result, right? So I need to learn about some of the syntax, just some basics of writing how to code. I need to understand the logic of how to find the right data and give it the right link. And then once I do, this console will check for me whether I'm doing the right thing. And now I can extend this to more advanced applications, like running a script to prepare a box plot, or maybe changing the way that this figure looks, right? And eventually I will be introduced to an assignment, and that assignment will be writing a complete piece of code, right? So if I know that I can complete this assignment, I'm not only following a step-by-step tutorial, but I'm in fact able to apply what I have learned. And now I can copy this and use it on my system to be able to run that every time I face a result. So what kinds of activities are possible for this type of learning? One of the training that we do in terms of SARS-CoV-2 is performing multiple sequence alignment and phylogenomics of the pandemic outbreak in different regions to really understand the emerging variants or clades that have been reported throughout the course of the pandemic. So as you can see, the logic of analysis is first done on the platform. And then we actually go through a series of steps to generate a variant report. And this is a report not unlike what people use when they use clinical reporting or when that reporting is being submitted to maybe public health institutions to understand whether there is a particular variant of concern, or maybe there are some number of samples that indicate that there is a spread of that variant of concern. And so we go through preparing such a report in R, how to install all of the different tools that are needed on your own system, and then how to actually run that with the specific types of variants that you might be interested in. So it really provides an ability to apply the logic, try it on publicly available data, and potentially then apply the same to novel data that is being collected. Now, we don't stay at the sequence level. We also try to incorporate the association between sequence and the structure. So we look at the spike protein, we understand the spike protein and even map the mutations and understand the relationship between the proteins so that we understand how they are interacting, and as a result, understand the significance of some of those variants. And all of that is done in under a month-long training program. Now, another big part of what we do, going beyond the traditional bioinformatics, where we deal a lot with sequence data and visualization, is actually learn about data science. And for data science, we have this very basic introduction that I just showed you in R and Python, but we also want to be able to apply some of those methods of exploratory data analysis, statistical analysis, machine learning in a more advanced environment. And so we created this program to offer greater insights into what is possible just knowing some of those basics. Again, this is project oriented. So we start with seemingly simple challenges. For example, in the context of precision medicine, can we identify a patient that is likely to respond or not respond to a particular type of medication? And then we use that information to be able to explain some of the additional downstream requirements for dealing with bigger data, how you can use different packages, how you can produce advanced visualizations, how you can apply similar approaches to different types of data, be it genomic, metagenomic, transcriptomic, and others, and eventually how to also apply this using machine learning, advanced statistical analysis, and an understanding of how to apply these skills to bigger data sets. Now, we've recently completed a program like this that was, all of it was designed around Python, from very basics to more and more advanced types of applications, where we also highlighted the significance of an online cloud-based environment to be able to write reproducible code. And I think those are two very important trends that students need to be introduced to as they move into the application of bioinformatics or data science to particular types of research. Not only do we need to be able to extract meaningful insights, we also need to know how we got those insights and provide an environment where others can easily reproduce the same code that we write, which means that the code needs to be readable, which means that we need to explain what we're doing and why we're doing that, and also highlight the results with a piece of code that somebody can take and run and get the exact same thing. And so there we cover things like dimensionality reduction, ordination and embedding. We look at different types of classification models, including the understanding of how those models work and how principles of those logistic regression, for example, methods are used in more advanced methods like deep learning, and also connect that with, again, practical examples. And those practical examples could be used for projects that have made a significant impact. So after we cover this domain of machine learning, we actually take a look at several different publications where all of the methods that we cover are explained in the abstract itself, and the student can really see and appreciate how the specific outcome from some research really affected the industry or even the clinical use of those methods. So for example, here, modeling precision treatment of breast cancer is a project that we use to illustrate individualized and personalized treatment, as well as predictive modeling to understand potential response that does not require data from that patient themselves over a course of different types of treatments, right? Which is obviously not possible. And I think very relevant to the medical field, which is moving further and further into the adoption of such tools and the precision treatment of different types of complex diseases. Now, beyond this training, and I'll be done here in a couple of minutes, we offer to use the same elements that I've just described, the T-Bioinfo platform for no-code bioinformatics, the coding elements that you can embed right into your coursework, your expertise in projects by illustrating a specific type of a method in application to a data set that you understand and might have published before, as a resource for researchers and faculty that would like to incorporate their own training and make it available to the NAIPI network. So one of the ideas is to identify gaps. For example, if you take an institution that might not have a full bioinformatics core, and they might not have the facilities to be able to support bioinformatics and computational biology research, we would like to match them with another institution that might have those capabilities, but might be lacking another capability that that university does have. And so by linking those, we're able to address the gap across the whole network. One example that I would like to show is a course that we designed together with Dr. Sona Vasudevan. She is the program director at the Georgetown University Medical Center for a program called Systems Medicine. And her specialty is in bioinformatics using 3D protein structures and really kind of looking at the relationship between sequence and structure. And so we worked with her to create a summer course where her students and students from other backgrounds could join and learn the basics of genomics in the virtual lab. We also worked with researchers that specialize in oncology, developing a program on precision oncology. And we also kind of design these together in collaboration with other researchers and faculty. And we are hoping to expand our network and really kind of build the same model of the context, the methodology, and examples of applications using curated data sets to expand the library of resources that we're offering. Now, we've done these programs over the several years now in several different institutions. We do have a lot of kind of feedback from different users, including students, faculty, researchers, and we are starting to work with industry as well, not only to provide the basic training and the path to research, but also for students that are interested in a path to an industry career, right? Where bioinformatics is a highly desired profession and skill set and can really make a difference in someone pursuing that direction. So thank you again for listening to my part of the presentation. It took a little bit longer than I anticipated, but I would like to pass this on to Dr. Mohit Mazumdar, who will speak about some of our ongoing programs that we have started in January and are now available to the NAIPI community for free throughout 2022.
Thank you, Ilya, and hello, everyone. It's an absolute pleasure to meet you all. Today, I'm going to talk about the opportunities, and I think many of you would be interested to learn about those opportunities, upcoming opportunities that you can participate in. So, is my screen visible to all of you?
Yes, it
Ilya,
is. Yeah, it is.
Okay. Okay, so thank you for that detailed overview, Ilya. And I think what we were trying to do is also to simplify this entire training using modular tools and intuitive multi-omics platform that you just saw. We are working with students and researchers, and we are providing research support and also big data analytics tools designed for biomedical data discovery. Ultimately, we are working for a future where we can contribute to enhance human health and well-being through biological research and discovery with the data solutions and support. And as you already saw during this presentation, what we are trying to do is that we are trying to close the gap for research and discovery by utilizing skills that are being developed throughout this training period with different training programs that are designed specifically to achieve some of the meaningful outcomes that we are highlighting, along with the application, learning about the application of these different new areas of research. So all of this starts with very basic. So it's very simple. It's just that we start from very basic coursework. Then we learn about these different data types. And then at the same time, we learn about how machine learning or data science or programming languages plays a role in downstream analysis and creating, better visualization and all of that. So this is the basic training that we offer. Along with that, there is a lot of project examples. So this is where it gets really interesting when you are being able to, apply these learning into some real hands-on data sets, do some real hands-on exercises, learn about through the practical examples. So Ilya did talk about all of this. many of these in quite detail, and I hope that you have gotten understanding. And please feel free to write in the chat if you have any questions throughout the presentation that you want to clarify. And again, about the structure, first of all, we are learning about the basic. We are learning about the different data analysis, different data types and their analysis. Then we are learning through example projects, and then we are trying to work on these specialization track projects. So these are high value research questions that we would want to answer in collaboration with the faculty or in collaboration with the student or a group. So about the steps, these are the different steps that we have. Very simple: introduction, then some specialization, and some examples to learn about the specialization, and then to be able to work on the specialization. And now I would like to tell you about the upcoming opportunity that we've been mentioning with the NAIPI network and with LBRN and LSU. So we are piloting. We are not piloting, we are just doing the spring programs. As we are doing spring programs and summer programs, really some exciting bunch of programs that helps many to start their bioinformatics journey, to develop these different type of skill sets. So we have different levels here: beginners level, intermediate level, and advanced level. So these programs will be available for many of you who would be interested to joining these mentor guided programs. So you can also see in this slide that there are a few dates that I put together, but we will go to the pages, the individual pages, where we have put together all the information, including the syllabus, the dates that have the asynchronous material that would be shared. So it has a combination of different elements, as you could see in the previous presentation by Ilya. He discussed about the courses. We discussed about, I showed you the project examples. So these have combination of all these resources put together as very innovative syllabuses in a meaningful way that lead you from basic training to different specialized training, and to be able to, start working on real data sets. So to tell you about the first program, Introduction to Bioinformatics, is just to get you started with bioinformatics. This online training is designed for everyone, including students who do not have any background in bioinformatics. So we will learn in this program, we will get an overview and learn about the processing sections such as genomics, transcriptomics for analyzing and interpreting omics data, and that will be followed by using machine learning techniques to interpret more insights from this data. So this is a basic program that would help many to get started. And in this program, we will also cover various public repositories. So how to find data, what are the different file formats that are that have application in biological research, and then it would be concluded by annotation, visualization, and interpretation of the results. So the entire concept and the entire idea with this program is to get everyone started, give them an overview of different sections that are available on the Bioinfo platform that you saw. Along with that, with every session, we will have asynchronous material. So there will be coursework associated with it. So bytes and molecules and introduction to bioinformatics course, then getting started with bioinformatics in R, getting started with bioinformatics in Python. And there are then 9 case studies that we will cover during this program. Now, I'll go into much more detail about each of these programs, and I'll show you that we have put together these pages that you can check and see the dates for your availability that will help you decide to join the program and get trained. And also, like these will be done through a mixture, like a hybrid program. There will be asynchronous sessions. Most of the sessions will be asynchronous, where we will be sharing with you a video and the online material, as well as the assignments and all the Google collab notebooks and all of that Ilya was just showing you. But here are some dates where we will have live sessions, and these will be at 1 p.m. CST time. But I'll again, we'll go to that in the next few slides. But before that, let me also tell you about the data science program. So in the data science program, which is again would be, we will be using Python and R, and we will learn throughout this course. Student will get an understanding of opportunities and limitations of machine learning in the context of preclinical and clinical research. This course is designed as a combination of online resources as well as the practical assignments and the live workshops. So all of these Omics Logic programs have these elements. And in this, throughout this course, we will review several project examples that demonstrates the successes and limitations of conventional machine learning methods and deep learning. And we will be using a lot of data sets that we have prepared for you to go through and understand like the challenges that we see when we use these methods, not only just to learn about the application, but what are the real challenges that you face when you start analyzing your own data, which is something that is not covered otherwise. So this will have a lot of sessions from feature selection, gene signature construction to regression and generalized linear models, network analysis, and as I said, deep learning, and then learning BioML and doing the coursework as well. So there will be also a specialized coursework associated with the data science program. Then we also have opportunity for you to join these programs, the basic data types, which is like to learn about genomic analysis. If you're working on genomic data, or if you have interest in working in genomic data, then in this genomic program, we will cover NGS genomics. So we will go through Sanger and WES and WGS, and then we will learn about the DNA code and the copy number variations. Then we will learn about the genomic sequence alignment and analysis, and we will try to visualize them through different browsers and popular tools that are available, not only through the Bioinfo platform, but the free publicly available resources. Then we will learn how to analyze genomic data using R, and then we will work on different pipelines that we have put together, like genome-wide association studies, and again, there will be a lot of case studies here. We will discuss special case studies from cancer, infectious diseases, and agriculture. So that would be about the genomics. And then there's also an opportunity to do metagenomic data analysis, to learn about metagenomic data analysis, which is really a field that is coming up and showing us those different type of associations that we were not even looking before. So learning about the microbiome, the functional bacterial community, 16S rRNA, metagenomic sequencing, ribosomal RNA, hypervariable regions, operational taxonomic units. I'm just discussing about the keywords because you will have an opportunity to learn about all of them in much more depth throughout these programs, and also to do not only just learn about it, but also practice this and run pipelines like QIIME2 data 2 pipeline for amplicon sequencing. Then we will look into the SRA and HMP and American gut project, human gut project, perhaps the viral genome detection through metagenomic data, looking at the evolution or fitness or quasi species. All of those things are kind of covered in this. We also look into the brain gut axis and how the gut microbiome affects by diet, and the effect of fertilizers to drug resistance. All those interesting projects are covered, and research ideas are being discussed in this metagenomic program. So that is a brief overview of all the upcoming opportunities that all of you have an opportunity to join, and I'll tell you more about these programs.
Sorry. So because it is almost time for this session, maybe you could.
So I'll just tell about the registration process.
Yeah, could you just paste the links in chat as well?
Right. So this is the link Ojasvi has already shared. I'll request Ojasvi to again maybe share this link. So please go to this link and you can fill this form that you can see on the screen and choose the program that you're interested in. If you're interested to join multiple programs, then you can obviously check 2 check boxes, but we will recommend you to do as per your availability. So you have options to check fill this form, and our team will reach out to you with all the information that we have discussed in this meeting and all the links that we have been discussing. We will also share these links with Luis, and I hope that Luis can also maybe help in sharing those links with the students who are interested and the faculty who are interested. But if you reach out, if you can fill out this form in this page on naipi.org training resources, and the second page, then we will be able to send you the details via email also. So please look for our emails. Many times this end up in spam or different filters. So please do check with some of these keywords, and you'll be able to see find the email in your inbox, and you will be able to learn and get all the links that are required. And we will be sharing with you the meeting links once you registered, so you will get access to different resources that we discussed today. And followed by these courses, one of the purpose of doing this is, again, to be able to learn about these different type of data types, and then to be able to work on a research project. So in the summer, we will be doing the research. We will be providing opportunity to all of you to work on your own research proposals. That would be high value research questions that you already are maybe working on, or that a question that you would be proposing after the training. That happens quite often. Many students go through this entire process and propose really amazing questions that could be easily that you can easily learn about, or maybe thought about, when you go through the data sets, explore the data sets, go through the publication. There are several ideas that keeps on coming that one would be interested to explore. So that opportunity would be available in the summer through the research fellowship program. And again, this program will have certain objectives that we would want to showcase as an outcome, to be able to get a poster presentation, to be able to showcase in different conferences, and to be able to publish also. So we also have a coursework associated with that, and we have several of these case studies and previous students who've been able to complete through this research fellowship program and showcase their results and publish their results. So all of this opportunity could be also through this Omics Logic Research Symposium, where we are bringing together students, faculty, researchers, and industry to discuss about recent advancements in omics data and bioinformatics. There will be a poster session, there will be prizes, there will be internship and career opportunities. All those things will be discussed in the research symposium. So we will be glad if many of you could also join this research symposium, which will be freely available to all of you who are interested. So that is what we kind of planned today to show all of you. Then we have a lot of research collaborations where we are working with different organizations who are providing us with mentors and additional support that would be helpful for many researchers who would envision collaboration in the future. And similarly, as I said, that these are our spring programs. Followed by that, we have our summer programs, where we have all these interesting programs coming up for you to take in the summer once you complete your basic training, and you'd be more interested, and it will be more relevant for all of you. So thank you all for your time, and I'll now pass it on to Ilya to conclude the session.
Thank you. Yes, I want to address some of the questions that I see here in the chat, but before we do that, I know that it's the end of the session. I see some people, Nicole, pasting some evaluation links. I don't know if you want to speak about that, or Jose, did you want to say anything? Okay, so there were two questions that I saw in the chat. One of them is to speak about what kind of data will you be able to access from your students, let's say if you offer this as a special topics course. So in fact, many people start with a special topics 1 credit course to give an overview. And what we provide is all of the activity data for those students. So I'll just, I already showed the profile, but briefly, you will be able to see all of the activity, which, for example, goes on to show what kind of assignments have been completed, what are the specific issues that the person faced, and all of this across the whole time. So you can actually go and check what was covered in that particular lesson. So there's a lot of activity, and all of this activity feeds into the types of courses that are completed, what those courses covered, as well as certificates for any of the coursework that were completed fully, that will also be available. So as a faculty, you will have access to all of your student data. So that was question number one. Question number two that I see, do you offer special assistance in your courses for Mac users? I'm a Mac user, so you can see here we have everything designed around online tools, first of all. So as much as possible, we try to avoid any of the technical limitations of different systems and configurations. That is typically what any trainer would spend 45% of their time is troubleshooting. And that's why we try to put as much as possible online. After that, we provide specific tutorials on how to install and configure your environment for Mac users, for Windows users, for Linux users as well. So if you have any specific questions, we can definitely help you with that. I think those were all the questions. One important thing that I want to clarify is that all of these programs are available for free for those that are members of NAIPI. The way we count members of NAIPI is if you are at a university or a research lab that is somehow funded through the IDEA program, then that makes it a part of NAIPI. And so if you can specify that in your application, that would help us kind of match you with that account. Thank you everyone for inviting us, and we hope to see you all again in the training that is coming up, and I'll pass this on to Jose. Dr. Medina.
Thank you, Ilya. And also Dr. Mazumdar, thank you very much for this very interesting orientation, informative seminar. I think we have lots of things to talk about and follow up on. And I want to ask our public, we have remaining in the chat 35 participants. I want to let them know that these orientations will also be repeated in some of our other network institutions. Luis asked me to mention that some of the Ana Jimenez institutions will be hosting this this orientation. So if you didn't get the full presentation, or if you want to just get a repeat, you can log on to the INBRE Puerto Rico INBRE. Just Google INBRE and you'll find the information for the next orientation here in Puerto Rico. So thank you so much, and we look forward to interesting collaborations with Pine Biotech in the future.
Thank you very much. And again, thank you for inviting us, and we will continue to look out for those of you that decide to participate. Thank you.
Okay, have a great afternoon. Thank you everyone for attending, and it's time to sign out now. So we'll see you soon in the next seminars.
Bye.
Bye-bye.
Yeah.
All right, thank you.